Hello friends, today we are going to talk about nucleic acid. First of all, you have to know about what are nucleic acid. In generally, you know DNA and RNA. Those are the nucleic acid. Nucleotide is the main part of the nucleic acid. It means the monomer unit of the nucleic acid. So, a nucleotide containing three types of components. First one is phosphate group and the other one is sugar molecule. You have to remember the sugar molecules only a monosaccharide and the other one is nitrogen base. These three type is called as nucleotide. In DNA and RNA, there are different types of structural, functional, and also the properties. It can be different. So, first in here, we have to know about the structural differences. In a DNA molecule and RNA molecule, these both two types containing a sugar molecule. The sugar molecule is a pentagonal sugar molecule. It means it has 5 carbon atoms. First, second, third, fourth, and other one is CH2OH group called. This is the fifth one. This nomenclature we have to number it. This carbon atom, not like other components nomenclature. In here we have to number this 1 dash, 2 dash, 3 dash, 4 dash and 5 dash. The main structural difference is the sugar molecule. DNA have deoxyribo sugar molecule and RNA have ribose sugar molecule. So, this is first carbon, second carbon, third carbon, fourth and fifth carbon. So, in here OH like upward or downward, it's okay. H, OH, H like this. In here, it can be H or OH group. If it a H group, we call it as deoxyribose sugar. If it is a OH group, it called as ribose sugar. This is the difference of the sugar molecule. So, if a deoxyribose, it has the H atom and in ribose sugar, it has a OH group. The next structural difference is nitrogen base. There are two types of nitrogen bases. First one is purine and the other one is pyrimidine. Okay. Purine and pyrimidine bases. In purine, adenine, guanine and cytosine. In pyrimidine bases, those are thiamine and uracil. In purine base, like this. Pentagonal and here a hexagonal structure. Those are This is a purine base and this is a pyrimidine base. Okay. First, a purine base. In this part, for carbon, 6 carbon, the numbering is 1, 2, 3, 4 and 5, 6. This is the hexagonal structure. And 7, 8, 9. So, for the adenine group, NH2 group present in 6th carbon. Guanine containing double bond O group in 6 carbon and NH2 group in 2nd carbon. Cytosine is the position change of guanine. And also in here, pyrimidine bases thymine and uracil. Little difference between thymine and uracil. Like this, a pyrimidine base H, double bond O and R, here also a R. If this R O, this R is a CH3 group, it called as thiamine. If the R is a H group, it called as uracil. Therefore, in deoxyribonucleic acid or DNA molecule containing adenine, thymine, guanine and cytosine, the RNA molecule containing adenine, guanine, uracil and cytosine. This is the difference between DNA and RNA. Nitrogen bases thiamine and uracil. Both pyrimidine nitrogen bases are different. In DNA contain thiamine in here and RNA contain uracil. So, this is the two structural differences. First one is the monosaccharide sugar and the other one is nitrogen bases. A DNA molecule containing adenine, guanine, cytosine and thymine. Between adenine and thymine, there is a one double bond. Between guanine and cytosine, remember there is only one triple bond. So, one double bond and one triple bond between adenine and thymine, guanine and cytosine. So, nucleotide is the attachment of phosphate group to carbon 5 is called as nucleotide. Okay. Now we are going to see what are the functions of nucleotide. First, acting as a monomer unit, formation of nucleic acid. And the other one is carries of energy required to metabolism reactions. Now we are going to see the attachment of a nucleotide. So, there are three components. What? Phosphate, sugar and also the base. First, you have to draw any DNA or RNA group, any monosaccharide like this. Okay. So, I am going to draw a deoxyribose sugar. OH group, this is the beta direction, H, and in deoxyribose it containing H. Here OH. So numbering of monosaccharides, the first carbon called 1 dash, others are 2 dash, 3 dash, 4 dash and also 5 dash carbon. In here, this is a deoxyribose sugar. So, for the first carbon atom in here, the nitrogen base is attached. In this carbon, the 5th carbon, the phosphate group is attached. Remember, we are going to draw only a nucleotide, but there are nucleoside also. We will discuss it later. So in nucleotide, this is the structure, and for this carbon also 1' carbon attached with a base, and here for a phosphate group. So this is, if there is a X, this is deoxyribose. If there is a OH group, this called as ribose. Okay. So we are going to see what are nucleosides. Nucleosides. What is a nucleoside? In a nucleotide, you know there are 3 components: phosphate group, monosaccharide sugar, and the base. But in here, a nucleoside containing only 2 components. This is the sugar molecule and also the nitrogen base. There are no any phosphate group. So nucleoside is like this: phosphate and the base. So remember, you can give a name, another name for the nucleotide. The attachment of what? Nucleoside plus phosphate group. Here nucleotide is what? Nucleotide plus the phosphate group. Okay. Previously we discussed about the structural differences between DNA and RNA. So in here we are going to discuss about the functional differences between DNA and RNA. In DNA, molecules can store all genetic information and it can be transferred in a generation to another generation. And also variations express genetic information can be do. So in RNA molecules, they cannot store genetic information and also they cannot transfer genetic information a generation to another generation. Both of DNA and RNA molecules involve to protein synthesis. So in my next video, I'm going to talk about the structure of DNA, complementary ratio, base pairing, RNA structure, and also the properties of DNA and RNA. Thank you for watching my video. Please subscribe to my channel. Thank you.